Hi everyone, this is Dr. Jiaxin Shen, a hematological physician from Shantou University Medical College. In the final session, the learning objectives for you are to differentiate blood groups and explain the process of determining ABO blood group clinically. Hopefully, you could then predict what needs to be considered during the blood transfusion process. Let's start with a scenario. One day, and you feel like doing something good, so you head over to your local blood bank, you get your finger pricked for a quick checkup, and somebody directs you to sit or lie back in a chair. Clean your inner elbow with alcohol. Put the tourniquet in your upper arm, and then insert a sterile needle into your vein. Once the bag is full, you get unhooked and grab a snack and drink to replace the blood sugar you lost. And the whole process takes around 15 minutes. Your blood will soon be taken to a lab, where it will be tested for infectious diseases, categorized by detailed blood type grouping, and separated into different parts before heading out to hospital. The first thing you need to know is that a patient who needs transfusion may only need some parts of the blood. Types of transfusible products that can be derived from blood include red cells, platelets, plasma, and cryoprecipitate, the protein needed for blood clotting. Before you give blood, you should also note what your blood type is. Do you have A, B, AB, or O? These different types all do the job equally well. They just represent different systems of your immunity. All the cells in your body have a plasma membrane which specialize glycoprotein markers on them that act like name tags or labels. These markers are your particular antigens, and your body's immune system is totally fine with that. But if it detects some foreign antigens, including those from viruses or bacteria, then it will send out antibodies to bind to those markers, often to tag them for destruction by the immune system. Your erythrocytes have specialized antigens on them, called agglutinogens, that activate antibodies which work by binding invading cells to each other, which causes coagulation or the clumping of blood. Which agglutinogens you have on your erythrocytes defines your blood type, and they are classified in two different ways. The first and the most important classification is by the type of agglutinogen, simply A and B. And your red blood cells can have either one, or both, or neither of these molecules. So the name of your blood type refers to what kind you have or don't have. Blood type A has A antigen, type B has B, AB has both, and O has neither. So why do you need to know what types before you give or receive blood? If you don't have a particular antigen on your blood cells, say the type A, then you have antibodies that will attack A-type cells should they enter your bloodstream. So people with AB type have both antigens and therefore no antibodies for either. Hence, they can receive A, B, AB, or O blood and are called universal recipients. Meanwhile, O-type individuals don't have A or B antigens, but have antibodies for both. That means that they can only accept other O-type. Type O cells don't have any antigens and won't be attacked if given to a person with blood type A, B, or AB. This is why a blood type O person is known as the universal donor. But just to complicate things a little bit more, you've got a whole other set of antigens with totally different protocol. There are your rhesus or Rh antigens named after the species of monkey where these antigens was first identified in. Much like A and B, you either have the Rh antigens, in which case you are Rh positive, or you don't and are Rh negative. Most of the population is Rh positive, so they don't have the anti-Rh antibodies, which means they can accept either Rh positive or negative blood. But Rh negative individuals should just receive Rh negative blood. And since the presence of AB antigen is controlled by genes different from that of Rh, we end up with 8 different blood types, 4 separate groups, each with 2 variations. Now we know that blood transfusion between donor and recipient of incompatible blood types can cause activated antibodies to attack erythrocytes and bind to components of the complement system to cause massive hemolysis of the transfused blood. Sometimes it will develop to even worse situation like renal failure, shock, and death. The figure on this slide briefly outlines the general procedure for determining ABO blood type, because it is so critical that blood groups are compatible. Cross-matching is needed. Cross-matching tests whether the recipient's serum will agglutinate the donor's RBC or the donor serum will agglutinate the recipient's RBCs. These tests make sure that the blood you donated could be safely received by someone who is really in need. Okay, to finally sum up, along the way, you learn the basic components of blood, including plasma, erythrocytes, leukocytes, and platelets. You understand these 4 elements in your blood come from one grandfather cell that is localized in the bone marrow. Along the way they became matured, they vary in chemical components and organelle structures. And finally, they are released into the circulation to carry out their respective functions. Also, we learn clinical cases and scenarios when these processes are flawed, causing various disorders. Additionally, we discussed the basic process of hemostasis that controls bleeding and how antigens are responsible for our blood type. Thanks for your watching, and many thanks as well to SUMC for those who make this course possible. That is all for the physiology of blood. Hope you can get inspired. Thank you.